Hi, everyone. So it is 11 o'clock and we are going to start our webinar now. My name is Kate Milliken. I have been a patient with the Corrine Dickinson Center for Multiple Sclerosis since 2006. I am thrilled to be hosting this webinar for the next hour. And we have a super full program. So I'd love to tell you exactly how it's going to play. Dr. Erin Miller will be sharing information from the European Committee for Treatment and Research for Multiple Sclerosis. This event is known as ECTRIMS. It's an annual conference. It's where a lot of the cutting edge research is presented, and it happened virtually in mid-October. So you'll be hearing a little bit about what's coming down the pike on that. Doctors Lublin, Katz-Sand, and Mahad will also be sharing the steps that they are taking at the CGD Center to treat patients who are living with MS and treating patients with other issues, how they are actually navigating this changing pandemic, and how our immune systems are responding to the vaccines. We will then open the floor to hear questions from you in light of what you've heard from us. So we'll be opening it live. We also have some questions that have been written in advance that we may tap into. So why don't we get started? I would like to start by introducing Dr. Erin Miller, who is the director of the Corrine Goldsmith Dickinson Center for Multiple Sclerosis. Dr. Miller.
Well, thanks, Kate, but just a correction. So I'm the medical director of the center. Dr. Lublin was, of course, the director, and he'll be speaking to you in a little while. I had the challenging task of distilling roughly 1,200 presentations down to a few to highlight in a few minutes. So I'm going to go pretty fast and just hit some high spots of things that I think that we at the center and you as people with MS or otherwise interested individuals. We're particularly interested in what's new in the way of disease-modifying therapies. So I think probably the greatest excitement in the field right now in terms of MS therapeutics relate to a class of drugs called the Bruton's tyrosine kinase inhibitors. And there are several of these drugs that are now entering or have just begun phase 3 trials. But also in cells called microglia and macrophages. And the preliminary phase 2 trials with a couple of these drugs have been positive in relapsing remitting MS, and these are now in phase 3 trials. But what's really exciting, because of the impact of these drugs on microglia and activated macrophages, is their potential for having success in progressive forms of MS, treating those stages of the disease. So there are now several phase 3 trials in both secondary and primary progressive MS. And at the Corrine Goldsmith Dickinson Center, we're involved in a trial of one of these drugs called tolebrutinib in both secondary progressive MS and primary progressive MS. The drug that's closest to approval that will add to our armamentarium is a drug called ublituximab, and the results of the phase 3 trial of ublituximab were presented at ECTRIMS. This is a B-cell depleting drug similar to two drugs already available, ocrelizumab or Ocrevus and ofatumumab, also known as Kesimpta. Ublituximab had remarkably successful results compared to another MS drug on the market called teriflunomide or Aubagio. People taking ublituximab had only roughly half as many relapses as those people taking the FDA-approved Aubagio, and they had dramatically fewer MRI lesions. So we look forward to seeing that drug on the market, I suspect probably within about a year. There's always a lot of interest among people with MS about stem cell transplantation. There's a lot of confusion among the public about what exactly we mean by stem cell transplantation, but one form is known as autologous hematopoietic stem cell transplantation, which you may also think of as bone marrow transplantation.
Hey, everyone, it appears that Dr. Miller lost his connection. While we wait for him to come back, maybe I can ask a question for Dr. Lublin. We were just beginning to talk about HSCT, the bone marrow stem cell transplant. I know there is a lot of confusion. And Dr. Lublin, maybe you could just comment in terms of the progress of it from what you've learned.
Well, so Aaron was about to explain the difference between the two different types of stem cell transplants. The one is bone marrow transplant, and that's designed to take the place of the disease-modifying therapies we now have, because it treats primarily the inflammatory aspect of the disease. The second type is what are called mesenchymal stem cell transplants, and those are putting in cells that will repair the damaged nervous system. At least that's the design of those studies. So they're very different approaches. We're testing the autologous hematologous stem cell transplant therapy now. It's never been properly tested. And we're doing it now because there are some major safety issues with that, because you're wiping out someone's bone marrow. And so it's not for the average individual. It's really for people who have failed our other therapies or have extraordinarily aggressive disease and just aren't responding. The prospect of repair through mesenchymal stem cell transplants is a fascinating one. We've done an early phase study that was reported out in ECTRIMS, and I suspect that Aaron was going to discuss. And it was a small number of individuals, only about 17 completed the study. But it was interestingly enough that it will move forward. And there's other approaches of that sort that are moving forward. And the idea of trying to repair the damage that's already been done.
Yeah, one of the things I'm aware of, certainly patients living with MS hear a lot about stem cell treatments that are international. And in light of what you know, when you kind of warn patients about that, would you say that those stem cell transplants are more of the second variety versus the HSCT?
I think they're more of the fraud variety.
Oh, well, there you go.
Okay, so anyone who has to pay for a stem cell transplant, that's fraud. It's just unproven, whether it be in this country or out of the country. The only way these studies should be done is part of a clinical trial where we can figure out whether they work and whether they're safe.
I'm sorry about that glitch. Zoom did what my wife has been unable to do, silence me. So I'll pick up. I think I dropped off when I was about to talk about depot glutarimer acetate. So many of you are probably familiar with Copaxone, which has been around for a quarter of a century and is given by 3 times a week injections. There is now a depot formulation that's been developed by an Israeli company, in which the drug is only given once a month by intramuscular injection. I've been involved as the principal investigator in a phase 3 trial of that agent that has now been fully enrolled, and results hopefully will be ready by next ECTRIMS. But there is also reported at this ECTRIMS meeting a very preliminary results from a very small trial of depot glutarimer acetate in primary progressive MS. There's been a lot of interest in the gut microbiome and in what's been referred to as syntax, the small intestinal network axis. There are connections anatomically between the gut and other organs, including the brain. And there was a presentation of interest. This was an experimental study in a mouse model of MS, in which one particular bacterium isolated from the gut, now I'm not even sure how to pronounce it, called Veillonella parvola, was inactivated. So these are not live bacteria, and they were given to the mice, and it resulted in lesser severity of this experimental model for MS. Of course, mice are not humans, and there's a long way till that. But that serves us as a segue into the question of diet and MS. And Alana who will be talking to you later, has had a particular interest in diet. And at ECTRIMS, she presented results of some of her work, which demonstrated that people who have better diets in terms of Mediterranean style diets with lots of fruits and vegetables, so those who endorsed better dietary habits had not only better quality of life, but actually performed better on a standardized neurological examination. We're always looking for better ways to diagnose MS and to offer more valid prognoses to patients. One of the things that's gotten a lot of traction in the last couple of years is something called the central vein sign. So MS lesions are inflammatory lesions that develop around a central vein. And these central veins can be imaged on commercially available MRI scanning. And the presence of multiple central vein signs is a good indication of MS. So sometimes when we look at MRIs, we can't be sure whether we're dealing with MS or some other condition, for example, a vascular disease or other inflammatory diseases. And the presence of central veins is very specific for MS. The problem is that it's you have to count a lot of lesions, which is not very practical. So at ECTRIMS, there was a presentation from a group of 10 MRI centers that looked at an automated method for identifying central vein sign. And that would be very helpful if that came to be fruitful. Another area that our center has had interest in, and this is a new addition to our center, Dr. Erin Beck, who came from the NIH, and she's particularly interested in better ways to image cortical lesions in MS. We know that we're very successful at imaging white matter lesions in brain MRI, but we're not very good at being able to identify the very many lesions that actually exist in the cortex. And she's working on that. But at ECTRIMS, there was a presentation that demonstrated that the presence of early cortical lesions at the very early stages of MS might be a predictor of a more adverse course, the development ultimately of secondary progressive MS. I want to turn now for a few minutes to cousins of MS, with which some of you might have an interest, and that is neuromyelitis optica spectrum disorder, and the more recently recognized disorder that now goes by the acronym of MOGAD, which stands for myelin oligodendrocyte protein glycoprotein antibody disease. And these are disorders that have similarities to MS, but respond differently to treatment and have different courses and prognosis. One paper at ECTRIMS highlighted the heterogeneity of the MOGAD presentations, a number of different clinical presentations, though most typically optic neuritis and transverse myelitis, which overlaps with MS and NMO spectrum disorder. One thing that's been found to be helpful in distinguishing between NMO spectrum disorder and MOGAD is something referred to as brighter spotty lesions. This was a paper reported from a group of Korean investigators in which they found that on spinal cord MRI, if you see lesions that are brighter than the usual lesions we see in the spinal cord in MS and in these other inflammatory disorders, the presence of these very bright lesions was very specific to NMO spectrum disorder and virtually never seen in MOGAD. A year or so ago, we had the exciting development that 3 new monoclonal antibodies were approved for the treatment of neuromyelitis optica spectrum disorder. These drugs, and you'll all be tested on whether you can pronounce them, eculizumab, inebilizumab, and satralizumab, are now on the market for the treatment of neuromyelitis optica spectrum disorder. And at this meeting, we saw continued evidence that these drugs continue to work in open label extension studies. We also saw data that indicates that MOGAD, which is sometimes refractory to treatment with other agents that affect the immune system, seems to be benefited by the treatment with intravenous immunoglobulin. And when you give IVIG, it seems that you can prevent or greatly reduce the frequency of additional relapses. Finally, I'll end by just saying a couple of words about teleneurology. Many of us were home for many months during the height of the COVID pandemic doing teleneurology visits. So there were several presentations about this at ECTRIMS, indicating that patients are very satisfied with teleneurology visits. And in one study, 65% of the patients queried preferred the teleneurology visits. Of course, that's probably because they don't have to take the time to travel, sometimes great distances. They don't have to take time off from work or studies to go and see the doctor. And another study demonstrated that care was not negatively impacted by the use of teleneurology rather than office visits. So I think we're already learning that teleneurology is here to stay. I don't think it's going to replace the office visit, but it can be used to supplement office visits. So I know I raced through a lot of information, and hopefully you found it useful, and I'll be happy to take questions. Okay, back to you.
We are going to do questions a little bit later, Dr. Miller. Congratulations on all those incredible words. I don't even know what armataria means, so I look forward to looking that up. I am curious for you, was there anything beneficial for you and your staff about teletherapy? I mean, was there something for you that was kind of a surprise in light of what you were able to learn about your patients?
So I think I learned during the 13 months that I was home doing nothing but teleneurology, which patients you can assess fairly reliably on teleneurology. And that's people like you who are normal, who have close to normal neurological examinations. Since most of our decision-making in MS depends on the history that patients tell us and the MR imaging more than the exam, we can make those decisions reliably with teleneurology. Where it breaks down is where we have to pick up subtle changes in the motor examination or the sensory examination. And there we really need to lay hands on the patient.
Yep, I hear you. Thank you very much for your presentation and finding a way to get back in the action. I really appreciate it. We are now going to move on to Dr. Lublin. Dr. Lublin is the Saunders Family Professor of Neurology. He is a director of the CGD Center, and he will be talking a little bit about COVID and the office. Dr. Lublin.
Thank you, Kate. So I will pick up where Aaron left off. And the things that I learned from telehealth is what people's little children and their pets look like, but both of which like the camera.
Aha.
So as Aaron said, with COVID, we switched almost exclusively to telehealth visits. And the transition actually went very well. We had a number of different platforms to use, some better than others, and we've gotten better with them. And of course, maintain HIPAA compliance. But the other thing we set up was an emergency visit schedule with specific faculty available to see individuals that had to come in, even though many people didn't want to come in and for good reason. So we did that. And the other thing that changed was we had redeployment of our faculty, our fellows, and our staff to COVID-affected areas of the hospital. And so now that we're getting back to whatever the normal is now, we are maintaining telehealth because, as you can imagine, with our center, people come from quite a distance. And it does let us do some of the visits for people who are far away more effectively. We're still waiting to see what legislation occurs to make telehealth more standard in terms of what insurance companies will pay for and what state licensing boards will allow. Now, in terms of COVID and MS, there's quite a few things that are worth noting. One was, in general, most of our patients who caught COVID did really quite well. The factors that we analyzed, and these match national, international standards, that were risk factors for doing worse with COVID if you had MS, were older age, obesity, and underlying disability, either from the MS or from some other cause. And those are really the same risk factors as the general population. In terms of flare-ups, we didn't really see COVID causing acute exacerbations of multiple sclerosis. What we did see was what are called pseudo relapses, which is an increase in symptoms that had already been there. And this is a well-known phenomenon known as Uthoff's phenomena, that when you raise body temperature, get an infection, a fever and such, old symptoms can come back because it has to do with physiology, but the efficiency of conduction through damaged nerves goes down when your body temperature goes up or your metabolism is thrown off, but not flare-ups. We haven't seen induction of MS following COVID infections. What we did see as part of the changes when we were treating individuals during the height of the pandemic here in New York was changing the timing that we utilize for intravenous infusions. So it's primarily those individuals who were getting Tysabri and those people who were getting Ocrevus and Rituxan, because there was limited availability in the infusion units because of spacing and patients not wanting to come in for good reason. And so we ended up with many patients spacing out the time between their infusions past what we normally would do. And that strategy worked out okay. And it's actually provided us some pretty good guidance as to what kind of flexibility we have in scheduling infusions. Now, the other issue that we have is, well, what was the effect of the immunomodulating agents that we use on patients if the risk of COVID and the severity of COVID? And there was a theoretical double-edged sword here. One was most of our medications, many of them lower your resistance to infection. We tell people this right away. There's a couple that don't, but most of them do. And so that was always a concern. But the other was severe COVID involved an upregulation of aspects of the immune response, a hyperactive response that Dr. Mahad can address further because she's an expert on this. And so theoretically, the medications we were using could have blunted that aspect of the more severe part of the COVID infection. And I don't think that's been fully sorted out. But what we saw for the most part, most of our medications did not affect the outcome of the disease. The only one that on a national and international level that has been an issue are what are called the anti-CD20s, and that's primarily Ocrevus and Rituxan. And the Europeans have reported more severe disease in individuals who were on those drugs. In a large US database, there also was a worse outcome in that group. In part, we personally have not seen that. At least I haven't seen that, and my colleagues comment differently. But nevertheless, that was the one group where there was a potential having done worse. Interestingly enough, and Dr. Mahad had predicted this at the very start of the infection of the pandemic, was that people on interferon tended to do better. And there's a good immunologic reason for that because interferon's involved in your immune response to fight off viruses. The last thing I want to comment on is vaccination. And so my colleagues and I have been extremely strong proponents of having all of our patients and everyone else around us vaccinated against COVID-19. For the most part, we have not seen reactions in vaccine affecting MS. We have not seen an unusual number of flare-ups after vaccination. Again, we've had people feel like their symptoms got a little worse, just like some people felt like they had a flu or body aches or such after the vaccine. Most of our medications have not had an effect on the ability of the individuals to get a response to the vaccine, except for the anti-CD20s, which would be the Ocrevus and the Rituxan, and probably also, we haven't looked at as much, another drug called Kesimpta, which works by the same mechanism. And there we've seen almost 2/3 of our patients don't get a response to the usual dosing of the vaccines. And now we're looking at the booster effects on that. And Dr. Katsan and Dr. Mahad talked about that more, and also talk about the fact that they still have some protection. The other group of agents were what are called the S1P agents, which are things like Gilenya and Zeposia and Kesimpta and Mayzent. And there was an Israeli study which reported a very poor response to vaccination with those agents. We found that about 70% of our people on those agents did get a response, but we're looking at that again with boosters as well. And the other thing that we've been spending a lot of time on is trying to figure out the best timing for giving the vaccines, especially in individuals that are getting like infused every 6 months. And we have some timing guidelines, how long after an infusion and how far before the infusion one should get the vaccine. So we've been working on this and working on the timing, and it's been kind of busy, but it's a good lesson for us for this pandemic, and it certainly will inform us for the next pandemic.
Yeah, great. Thank you so much, Dr. Loveland. Instead of asking you a follow-up question right now, I want to roll right into Dr. Katzand, because speaking from my world of the patient population, I think this matter of trying to weigh an MS medicine with a vaccination and deciding which one is more important is pretty intense for the people who are on these specific drugs. So Dr. Katzand is an associate director from the CGD Center. And Dr. Katzand, why don't we talk a little bit about your study, which ties in nicely with Dr. Loveland?
Sure. So I'll tell you a little bit about I'll build upon what Dr. Loveland was talking about.
Thanks.
And tell you a little bit about the work that we've been doing so far, and then, why we decided to do this study, which we're calling the booster study, but is, as of right now, a third injection study, is not quite a booster in people who have an immune system issue. So as Dr. Loveland said, we haven't really seen, severe outcomes in our patients over and above what's been seen in the general population, maybe a little bit with people on B-cell therapies. But of course, we want our patients to be as protected as possible. And since what we've seen has been consistent with what's seen in the general population, that does mean that we have had people in our practice who have died of COVID. Also, as Dr. Loveland said, we've all had patients who, after having COVID, continue to have symptoms months and months afterwards. I think it's more than just pseudo relapse. I think because some of the symptoms of like long COVID overlap with some of the symptoms that our patients already experience, things like fatigue and mood changes, which can already be issues for a lot of our patients. I think having COVID can be particularly problematic for people. So of course we'd like to avoid it if possible. And so that brings us to the issue of vaccines and how well vaccines can protect people who are on disease-modifying therapies. So the work that we have done so far shows that for people on most disease-modifying therapies outside of B-cell therapies like Ocrevus or rituximab, and outside of the S1P modulators like Delenia or Mayzent or Zeposia, people on all of the other medications like Tecfidera, Tysabri, all seem to have had good antibody responses to the vaccines. So we actually looked at a cohort of people at our center who had clinical antibody testing done, and nearly 100% of them showed a good antibody response to the vaccine. People who were on B-cell therapies did not universally show that. So actually most people did not. So about 65% of our patients on a B-cell therapy had no clinically detectable antibody response to the vaccine. Now, what does that mean? The problem is we don't know what that means. We know that antibodies are not everything. In terms of thinking about the immune response to this virus, there are a lot of other parts to the immune system that are responsible for responding. It's not only the antibodies, right? The problem is that from a clinical standpoint, when I have someone sitting with me in the office and someone wants to know, did I have a good response to this vaccine or not? The only test I can order that's available in a clinical lab is an antibody test. So we've used antibody tests kind of as a proxy. I think what antibody tests that are available now can tell us is that if someone has a good antibody response, we think it's a very good correlate to show us that the rest of the immune system should also be responding, unless they have some other specific, immune problem with the rest of their immune system, which most of our patients don't. The problem is that when the antibody test is negative, we think there's still a very reasonable chance that people have an immune response to the vaccine. It's just that, we don't have a way to measure it. And that's where Dr. Mahad and her team comes in, and that's why we're doing the study that we're doing. So I'll tell you a little bit about the structure of the study that we're doing. So we are drawing blood from people who have gotten their initial vaccine series and are about to get their third injection, as per current recommendations for people who are immune compromised, which are patients who are on Ocrevus or on an S1P medication like Gilenya meet those criteria. So in people who are about to get that third injection, we take a blood sample and we look not only at antibodies, but we look at we do this really sophisticated testing with Dr. Mahad's lab, looking at other components of the immune system, for example, the T cell responses to the vaccine. So we'll look to see at baseline before that third injection is done. We'll get a kind of a good sense of where people actually were after their initial vaccine series. Then they go ahead and get their third injection. And then we have them back after 4 weeks to look at what is the response at 4 weeks. And then we have them back again at 12 weeks. So we can see what is the response at 12 weeks. We don't know what the trajectory is going to look like. We don't know if people are going to maybe mount a response, but it's going to take longer than the 4 weeks, in people who are immune competent. We don't know if that's going to be the point of maximal response, and that by 12 weeks, maybe the response is not going to be as durable in these patients. We really don't know. And that's why the study is so critically important. I'm excited to say that as of today, we've enrolled 40 participants in this study. And I've asked our colleagues to try to get us some more by talking to their patients about how important this topic is. And, I think it's tough because, as always, we're asking people to do something active to participate. And here we're asking them to come in for 3 blood draws.
Yeah.
So, we're trying our best to make it convenient for people and try to bundle the lab draws with an infusion visit, with an MRI visit, a neuropsychology visit, an office visit, so that people are not having to make extra trips. And so I think, and our research team has been extraordinarily accommodating about coming in early if someone wants to come in before work, before the lab is open, to meet people to try to make this work. So I'm really hopeful that we can get our numbers up even higher, because the more people we enroll, the more we're going to be able to learn about questions like the one you posed about timing, for example. Right?
It's funny.
The more people we get who have different, timing of their vaccine with relationship to their medication, the easier it's going to be for us to tell whether that's important, which is a huge thing right now that we're trying to figure out.
Sure, because you want them to have both. You want them to be as safe as they can. And what's amazing about this COVID experience for everybody is an element of it of figuring it out in real time. And I do believe this partnership with Dr. Mahad, who you guys are all about to meet, is incredible. So let me introduce Dr. Mahad. She is the director of Precision Immunology Institute, also the director of Human Immune Monitoring Core at Mount Sinai. Dr. Mahad, would love to hear about this partnership and some of the stuff you work with.
Yeah. So indeed, it is the beauty of this Mount Sinai institution, this integration between discipline, because this is where discoveries and finding and progress happen. So I am an oncologist by training. I'm also an immunologist. I lead the Precision Immunology Institute, which goals are really twofold. The first is to study the basic of an immune response, the basic of an inflammatory response. And there is still a lot to learn. The immune system is a very sophisticated system that has been shaped during evolution to respond to threat. And there's many, many things that we still have to understand. And the reason why we need to understand it is because we can use it then to develop novel immunotherapy strategies. So this is the second goal of the Institute, is really to develop novel immunomodulatory strategies to dampen inflammatory disease, like in MS, and also or increase immune response in cancer patients, which is the focus, one of the focus of my laboratory. So, but also we think about novel immune treatment, so enhancing inflammation, dampening inflammation, but also vaccine is part of that. Vaccine is how you can really mount a response to prevent infection disease, and we are doing also a lot of vaccine work in cancer. So when I also founded, just because it's important for the rest to understand the partnership here, we also founded through the Institute this Immune Monitoring Center. And the Immune Monitoring Center is really a place where we have coalesce a lot of technology. You know, there is a big revolution in technology development that allows us for the first time, I think, in the history of medicine, to look with a lot of granularities are at the cell per cell basis, looking at the protein content, the RNA content. I don't know whether these terms are familiar to you, but it's really looking at the composition of this immune response with much more granularity that we have ever been able to do before. So, but this requires that we have very sophisticated instruments, that we have staff to run it, and that we incorporate this type of measurement in the clinic. These are not simple tasks, and this is where we partner with Dr. Lublin and Dr. Katsan to really think of how we can really harness this development in technology to learn about the disease and learn about responses to treatment, or learn how to develop novel therapy. So when COVID hit, one of the big questions we had, the first question we started to address is why this COVID disease was so heterogeneous, why some patients were developing the mild disease and some were dying from the disease. And so what we started to do is use that technology center to measure the inflammatory response and the immune response, and also measure the virus in tissue and virus proliferation. And what we quickly realized is that patients were not dying of virus proliferation, or virus taking over the organs, but patients were dying of inflammation. And that we realized that was the case. We realized that quite soon. And in fact, patients were able to mount a response against the virus, block virus infection, and that it still progressed. And some end up dying despite not having a single virus left in their body. So then the big question started to really understand why some develop this very dramatic inflammation. And this is where it becomes very important to understand whether patients on immunomodulatory strategies were protected or were at risk. And we started a large effort to understand that. And for example, we realized that patients on anti-inflammatory drug like steroid were at risk, because when you block the entire immune response, you cannot mount an antiviral response. But when you had targeted immunosuppression, like for example, TNF blockade, you could be protected from very severe COVID-19. And this is where we hypothesize that having the right dose of interferon, which is an antiviral drug, could potentially prevent this virus to continue to expand, and this will prevent the trigger of this big inflammatory disease. And then what we started to do very quickly after started to study the acute infection, we started to study and focus on patients that developed this post-COVID syndrome. Some patients had these lingering symptoms for very prolonged period of time. This is a big focus of the center now, is to understand why some patients have this symptom that don't seem to resolve. And we have several hypotheses to explain why this happens. In fact, we have progressed also a lot in understanding why patients develop this very severe acute disease, and I'm happy to discuss some of these findings. But then now what interests us with Dr. Lublin and Dr. Katz is to understand these patients of ours, MS patients that are on some different type of immunotherapy, and whether this immunotherapy now may compromise their ability to respond to vaccine, because anything that somehow modulates the immune response can affect our ability to respond to an immune challenge, which is vaccine in this case. So what we did, again, we took the center and we used this high-end technology to understand with great detail the response to first the vaccine and potentially the booster, and look at it in different group of patients that receive different type of therapy. And in most laboratory at Walgreens or in most places, you can measure antibody to one or two protein. This is just one little aspect of your immune response. The immune response is very wide. There are many ways of responding. Even the antibody you can have a very specific and focused response to one part of the virus. You can have a larger spectrum of antibody. There is another arm that is quite protective, which we call the T-cell arm that Dr. Katz discussed. We are measuring all of these in these patients, and in the study that Dr. Katz mentioned, we are quite excited about the study. The goal now is to be able to do all these assays. What we want, and she's very ambitious because she asked that we don't compromise on anything. And she wants us to look at all the aspects of the immune response prior to the booster, a different time after the booster, so that we can comprehend really how the treatment that our MS patients are taking compromise or doesn't, or compromise one aspect of immune response. And this is what we are doing.
Wow. Dr. Mahad, first of all, I want to apologize for mispronouncing your name. This is super exciting stuff. And the thing that I hear when I actually hear that you're beginning to notice different trends or different variables, what happens now? Because ultimately a patient is listening to this. It's very easy to get excited about this work. Yeah. But when you think about how it's going to help somebody in the future, how do you sit with all these moments of kind of small triumphs, and what happens next for patients? Yeah.
So the first, I think, question we are asking with Fred and Ilana is, are a booster useful? So it's going to be very simple. We are going to measure the response before and after and see whether we are broadening or expanding this immune response. We also have a way of measuring whether that response that we are developing pre or post booster it can block the virus. Block virus ability to infect cells. We have a little in vitro system. In vitro means a bench, a little Petri dish where we put a virus and a cell. So we mimic what could happen, in an individual. And we take those antibody or those T cells and we say, are they efficient? Are they protective, right? Mm-hmm. Then if we find that this is not the case, then this is where we have to come up and discuss, right? Should we add another booster? Should we be thinking about different vaccine? Right. Because we know that when we change the vaccine strategy, we can affect different arm of the immune system, but we need to have those measurements. Remember, in the US, we mostly used mRNA vaccine. mRNA vaccines are very good to induce an antibody response. It's not clear yet what is the strength of the T-cell response. So there is a lot of subtlety here, but definitely having this measurement will enable us to say, okay, what's the next step? Should we go to another? Should we boost with another vaccine or not? And there is a lot we can do, but we need to get the data first.
And I think also, Kate, with regard to this question and your earlier question about timing, this also, I think, will be informative for us in terms of timing of medications, because, based on our early data, we've recommended to our patients that if they're on a B-cell therapy that they get every 6 months, that they wait until they're at least 3 months past the infusion. But if they get closer to 6 months, it's probably better, but now we can actually learn that, because if we can enroll enough people who got it at different time points, we should be able to see, the efficiency of the response and how much it matters. Some of our colleagues also recently published a study showing that people who had their infusions delayed during the pandemic seem to have done okay from an MS standpoint. And so in terms of, that helps us in terms of thinking about how to balance potential, risk and benefit. And, we think if we show in our study that people who waited a little bit longer in terms of timing of the infusion to get their vaccine and push their infusion out a little bit, if we find that that's a more efficient way to do things from an immune perspective, maybe that's the strategy that we do with our next, round of boosters. So I think we can learn a lot about this, and we're trying, as I mentioned, to enroll the study as quickly as possible so that we can get back to people with data, you know, in real time. This is not something we want to find out 2 years from now. This is something we want to figure out like in the next.
Right, and you have the ability in this time, if you do come up with something that may be helpful, to express it and use it moving forward, even if it's not a 2-year process of rolling it out more formally.
And I do want to mention that we are very grateful to a donor family who is supporting this work.
Wow.
We are very grateful for that, because if we want to be able to do things in real time, it's not the kind of thing where you can say, okay, we're going to write a grant, we're going to submit it, we're going to wait 6 months for someone to review it and then get back to us, and they want us to revise it. And, you know, most of the time when you submit a grant, I think some people know this, you know, it takes, you know, 1 to 3 years to get something funded.
Yeah.
And that's not something we want to be working with right now.
So certainly thinking about this time of COVID being a very stressful one, there are a lot of things that have happened in your world that have been accelerated. I mean, I think in the healthcare space, thinking about telehealth, a lot of people kind of got kicked off the ledge of having to use it, and here we are. So what are some other things that you feel have been really positive in terms of progress during this time at the center?
One of the things is the flexibility, the potential flexibility in dosing schedules that Alana just related to, and we have some data, in fact, that was generated with our former colleague Eliza Ben-Zachariah on being able to delay things like Tysabri infusions. So we did that on the cuff during the things, but she and others have developed some data to suggest that this, in fact, can be done more routinely. The same thing happened with the B-cell inhibitors, where we were able to delay them. What we need to do in that case is develop the same kind of metrics that we did with Tysabri to make sure that we've looked at the safety and efficacy moving forward. The other thing is it reinforced for me the resilience of my colleagues. To be able to perform under exceptional, severe, challenging circumstances, and also being sent to areas that were well outside their comfort zone and taking care of some very sick people.
Yeah. Dr. Miller, anything you'd like to add?
You're muted.
Sorry, can you hear me now?
You can. Yep.
So I think one of the things that we had to focus on with teleneurology is doing it safely. I was involved with the National MS Society in developing some guidelines about doing teleneurology, and I heard some stories of some unfortunate events that happened during teleneurology. I had one experience myself where an older woman fell when I was testing her gait. She was alone in the apartment. Fortunately, she had a good connection with her doorman and the staff of the building, and she wasn't injured, thank goodness. And they came up right away. But we need to know what the circumstances are when we're examining somebody who may be home alone, for example.
Right. Well, I appreciate the idea of things coming out of this that are surprising and helpful that maybe you weren't initially thinking of testing or realizations. I think that's one of the things that's so great about philanthropy is having an idea, starting to make progress on an idea, finding additional insights. I would love to open the floor to some questions now, because we have about 10 minutes remaining. Anybody who is listening and would like to ask a question, there is a Q&A option on this webinar at the bottom, kind of where your doc is. Please feel free to put out a question, but I'm going to ask a couple that have been written in advance. One of them is this: will switching from Pfizer to Moderna or vice versa stimulate an immune response? I've had 3 doses of Pfizer and I still do not have any antibodies. Anybody want to take that on?
Miriam, you want to try that one?
So the first question is the patients on immunomodulatory agents? So I suppose she's probably on B-cell targeting agents. Seems right, Fred, that this is
Yes.
what this
Assume
patient
that.
So we are assuming this. So this is a very good example of the patients we would like to study in our cohort, right? So she received the booster, it seems, or she or he received the booster and cells. So the first thing I will do is look at the T cell response. Are they developing any type of T cell response or not? And then the next step would be, so we switch to a viral vector, and this is, I think, a very valid discussion to have. So the patients receive a viral-based vaccine to broaden up a little bit.
So that would be like the J&J vaccine.
Yeah, J&J vaccine. The other possibility, and that my colleague should discuss that, is the possibility to reduce maybe the B-cell targeting agents so that vaccinate, so that they mount a vaccine response. There is an antibody that circulate and maintain, and then my colleague could discuss that. But that's also
I mean, this is what we've been talking about in terms of potentially delaying the next infusion, and hoping that will get us a better response. And we're going to get some data on that pretty soon. Yeah. But it's a really good question. There is a trial that's going on that I saw, and I think it's our colleague Dr. Baror who's the MS arm, but I think it's going on in multiple autoimmune conditions where they are actually randomly assigning people for their booster, different injections. Mm-hmm. And so I think that'll be a helpful. It's a small study, but I think that'll be helpful for us in terms of seeing. We'll be able to look at that from an observational perspective, because in our study, we're having people who are, we're entering people who are getting which booster. And so, but having a clinical trial that actually randomly assigns people to get this booster or that booster, I think is going to be really helpful at figuring that out too.
Right.
It is, I think, important for people who don't have antibody responses, and while we're trying to find out what their protection level is, to be aware that antibodies can be administered passively, and so if you get sick and very early on you get an infusion of monoclonal antibodies, you will likely have a lessening of the severity of the disease.
And I might add that we and just about all of our international colleagues have had the view from day one of the pandemic that not forget we need to treat MS first and then think about how the pandemic is affecting that. But MS is still a disease that we need to make sure we have carefully treated and adequately treated.
I am going to reach out to the person that asked the question. Maybe we can get them in your trial. That's great.
Yeah.
Oh, okay. Here is a question that just came in as a result of what has been heard. Do we know anything about whether the impact of vaccine immunization of the B-cell agents is retroactive? So if you weren't on one of those meds, but later go on to one, does it impact immunity that you've potentially built?
That's a really good question. Less so. It's less of an issue. That came up several times in our study in terms of the coordinators coming to us and saying, oh, we have someone who we think is eligible, but they weren't on it for their initial vaccine series and they're on it now. And I said, you know what, let's enroll them. It's not exactly how we plan to do it, but I think we have 3 or 4 people in our study now who meet that category, because we're going to have more and more people like that, right? As this goes on longer and longer, we're going to have more people who were on no therapy or on different therapy when they've got their initial vaccine series, or they had COVID or whatever. And then as time goes on, they go on to one of these therapies. And then what's going to be the response from there? It's easier for your immune system to maintain a response that it's had before going on to one of these medications than it is to respond to something totally new, right? So in our patients who go on to Ocrevus, having had many years behind them of flu shots, for example, or having had the flu or whatever, it's very different than looking at a COVID vaccine where they have no prior, immune memory for this. So yes, we do think it's easier. I don't know, Dr. Mahad, if you wanted to add anything to that.
No, so I understood the question whether whether B-cell targeting agents can somehow affect the antibody level. No, I think this is what the patient is worrying about. So it's unclear. I think it's unclear how it does affect some cells that produce antibody, but likely not a very, so there's different level of cells that produce antibody, right? It's a little bit complex and we still need to learn, but I will agree with Dr. Katz is that it is, I will feel, I think it is, it appears potentially judicious to reduce the B-cell targeting agents, vaccinate, have a good level of antibody, and potentially put someone back on B-cell targeting agent, and then following, the antibody level. It seems to us that it's likely that the B cells the antibody could be maintained. It has to be studied in more detail. But there is a lot of reason to think that you can maintain an antibody level for some time, despite the fact that you are on a B-cell targeting agent.
Wow. Okay, next question. A number of reputable MS clinics, including UCSF, now routinely recommend extended interval dosing for Ocrevus. What is Mount Sinai's perspective here, and what's the longest interval between infusions that you're comfortable with?
So there's a couple of philosophical issues there. One is that there is a marker for what the Ocrevus is doing, and that is you can measure B cell levels. And so theoretically, one could dose based on the B cell recovery. We haven't done that just because that's not the way it was labeled and done. We do that to some degree in neuromyelitis optica, where we really don't want any B cells to return. In MS, it's not as clear what level of B cells you need to maintain to keep the therapeutic effect. But that it is a strategy. I think there's a study that's looking at that now to see how it works out. We don't really have data yet.
How probable would it, I'm sorry, Dr. Miller, just really quickly, how probable would it be to actually measure B cells?
Oh, it's easy. We do it all the time.
Okay. Dr. Miller?
Well, two comments about that. First of all, I suspect that question really was referring to extending the interval between doses of Tysabri rather than intervals of Ocrevus. I don't really believe people are routinely extending the intervals for Ocrevus. But we don't really know how important those B cell levels are. I just saw this morning rather, a patient who's been on Ocrevus for 4 years. He has not had an attack or a new MRI lesion during those 4 years, and yet by 4 months he has plentiful B cells present on a couple of recent blood draws. So we don't really know how low your B cell levels have to be and for how long after these Ocrevus treatments to make a difference. And maybe Dr. Mahad wants to comment further about that.
I agree with you guys. I think it makes a lot of sense. Yeah.
Okay, I'm going to ask one more question, which has also just come in watching this. Have you seen increased rates of long COVID for people on B cell depleting therapies?
No. And I'm pretty sure the international database did not see that either, or the national database.
On the contrary, I will say it could be beneficial. Because one of the possibilities that this long COVID may be due to increase autoantibody development due to this big inflammation. So you make antibody against your own tissue, kind of this inflammation leading to an autoimmune disease. So this is one of the strongest hypotheses right now. And it seems that they may benefit from B cell targeting agent. Definitely IVIG will be tried in some patients. And so we'll have more data coming soon, hopefully. Sinai is a big center for long COVID research. We received a very large grant from the NIH to study thousands of patients with long COVID syndrome. So hopefully, this will shed some light on the mechanism of this disease.
That's great.
I think the issue is more, as we were saying before, that there's some overlap, of COVID symptoms with MS symptoms. Sure. In terms of long-term symptoms, and that after people have COVID, people who have MS and have some of those symptoms already can take a longer time to recover. I think that's probably more what we're seeing than the immunologic phenomenon that Dr. Mahad is describing.
Got it. So we have a couple more questions. We don't have time for them, but for the people that ask those questions, we will be in touch with responses for you. I would just like to thank everybody who has been here for the whole hour and these amazing panelists for giving us this information. I'm hopeful you all who are watching feel smarter and more empowered and certainly inspired that there's all of this stuff happening, not only in the world, but here at the center and Mount Sinai. So we're going to put up a flow code that will give you an opportunity to donate to the center. Certainly, philanthropy is hugely important in all of these studies. And actually, there have been a number of examples of wonderful things being proven, even if they weren't the primary objective. So I hope you guys will all consider, who's watching, donating, because the work's only begun. And we do need your help to give these talented leaders the resources they need to keep going, to keep innovating, and to keep bringing innovations into this world. So this is a QR code, and all you need to do is point your smartphone camera at it, and it will take you to a web page where you can support this urgent work. I really enjoyed being here. Thank you all for tuning in, and stay safe.
Thanks.
Thanks, everyone.
Thank you all.
Thanks.
Thanks, everybody.